Dear students, welcome to the program. Today we'll be having another important topic on mushroom cultivation technology. Our today's topic is entitled as Casing and Casing Materials Used in Mushroom Cultivation. This topic is very important from the perspective that the mushrooms require a microenvironment and fruiting initiation. Let's have a look on today's objectives. Our first objective will be to have a detailed introduction about casing and casing materials used for mushroom cultivation. Our second objective will be to make the students aware of the physical, chemical and microbiological factors of the casing layer, and our third objective will be to have a brief discussion on functions of the casing layer. Our fourth objective will be to acquaint the students with the choice of casing material used, and our fifth objective will be to discuss briefly the preparation and application of casing material, and finally we'll be having concluding remarks on today's discussion. Dear students, let's begin our topic with the introduction. Mushroom cultivation is a delicate process that requires precise environmental control to ensure optimal growth and fruiting. One of the critical steps in this process is the application of a casing layer over the colonized substrate. Casing refers to a non-nutritive top layer that provides a microclimate favorable for the development of mushroom fruiting bodies. It serves several essential functions, including moisture retention, protection of the substrate, and stimulation of mushroom fruiting. The choice of casing material is crucial as it directly influences the yield, quality, and health of the mushroom crop. Common casing materials such as peat, moss, coco coir, and vermiculite are selected for their ability to retain moisture while providing adequate aeration. Proper preparation and maintenance of the casing layer are necessary to create the ideal conditions for mushroom growth. Understanding the role of the casing and its materials is fundamental for mushroom cultivators aiming for high productivity and quality yields. Among the stages of mushroom cultivation, the addition of the casing layer is essential to achieve high productivity. The casing layer is the material used to cover the mushroom compost after complete colonization to induce the transition from vegetative to reproductive growth. However, it should be noted that this step is not mandatory for all mushroom species, being essential for agaricus mushrooms and optional for Pleurotus species and Coprinus comatus. The induction of fruiting is not only genetically controlled but also strongly influenced by environmental factors. Due to the large differences between the physicochemical properties of the casing layer and the compost, covering the compost with such a different material triggers a transition from vegetative fungal growth to the reproductive process. The casing layer therefore promotes an ecological change that is decisive in this transition and is as important as the quality of the compost and the management of cultivation conditions. Thus, the induction of fruiting depends not only on genetic capacity of the mycelium but also on physical, chemical and microbiological factors. In this context, the casing layer is one of the most important aspects of mushroom production, influenced by these factors in addition to the important parameters of temperature and relative humidity. Peat is the most widely used casing material for the cultivation of agaricus mushrooms, mainly due to its water retention capacity and porosity. However, good quality peat, in addition to being subject to legal restrictions, is not available in all countries, and peat is not a renewable material. Therefore, the search for alternative materials has become very important. However, these materials must have appropriate microbiological and physicochemical characteristics for both good colonization and an adequate response to fruiting induction. Some of these factors will be addressed as follows, as well as the types of materials that can be used to replace peat. Dear students, let's now talk about the physical and chemical factors of the casing layer. Physical and chemical factors such as particle size, electrical conductivity, and hydrogen ion concentration, that is the pH, are considered determinants of the quality of the material used as a casing layer. Particle size has a direct effect on the compaction capacity of the casing layer and consequently on its water retention and aeration capacity. If the casing layer has high electrical conductivity, mushroom fruiting is inhibited or drastically reduced. Regarding pH, although fungi can grow in a slightly acidic environment, the casing layer should have an alkaline pH of up to pH 8. The use of neutral or alkaline pH in the casing layer is important not only for the physiology of the fungus but also to help control the presence of competitors such as trichoderma, which grows better at acidic pH. Due to all these factors, materials are rarely used in pure form, being combined instead with other materials or corrective materials to achieve desirable properties in the casing layer, especially in terms of pH and porosity. After complete colonization of the casing layer, fruiting induction management should begin. In general, fruiting induction depends on three parameters: temperature, ventilation, and relative humidity. Of course, the interactions of these factors with the casing layer determine whether effective fruiting occurs. For this reason, none of these three factors can be neglected in studies on the casing layer. Thus, the relative humidity of the air and to a large extent ventilation should not vary substantially from one species to another in commercial crops, except in some special cases. Temperature is therefore the factor that should undergo the greatest variation, especially for mushrooms that depend on cold conditions for fruiting. There is no exact temperature for the induction of Agaricus bisporus fruiting. However, values between 15 and 17 degrees Celsius are usually used. According to Covey, 1972, the ideal temperature should be 7 to 9 degrees Celsius below the temperature considered optimal for substrate colonization, which is around 22 to 25 degrees Celsius. This temperature difference is due to ecological issues. Agaricus bisporus is a species originating from a temperate climate in which the seasons are well defined. In nature, this seasonal definition causes the fungus to multiply vegetatively during the hot season and start to fruit in autumn. Therefore, an ambient temperature of 22 to 28 degrees Celsius is suitable for the colonization of the substrate and the casing layer, but it is not suitable for the induction of fruiting. According to Flegg, 1989, fruiting can occur without reducing the temperature. However, the resulting process would be suboptimal. After the induction of fruiting, the temperature can be returned to 25 degrees Celsius, which can speed up the mushroom development. However, the maintenance of a low temperature also helps in the control of pathogens and contaminants. Fruiting usually also requires high relative humidity because basidiocarps do not have defense mechanisms against dryness. When substrate colonization occurs inside bags, there is no need to maintain such a relative humidity. Humidity restrictions change when colonization is performed in mushroom beds, in which the substrate is exposed to the environment, or when colonization occurs in perforated bags. In these cases, it is essential to maintain high relative humidity. According to Carrasco et al. In 2021, the following relative humidity standards should be followed for the cultivation of Agaricus bisporus in a bed system. A. It should be greater than 95% until fruiting. B. It should be around 90 to 95% during fruiting. And C. It should be 80 to 85% during mushroom development. Regarding aeration, it is necessary to reduce the concentration of carbon dioxide in the air, since carbon dioxide is extremely harmful to the development of mushrooms. Although the fungi are quite tolerant to carbon dioxide during the colonization phase. For fruiting induction, the concentration of carbon dioxide in the air must be held at 800 to 1000 ppm. Evidently, the casing layer plays a very important role in this change in carbon dioxide concentration in the air, since more porous materials will be more favorable to the process. Therefore, the ideal porosity and water holding capacity of the casing layer must be balanced. Dear students, let's now talk about the microbiological factors involved during mushroom cultivation. The importance of microbial interactions for the fruiting of mushrooms has been documented for many years. For mycorrhizal mushrooms, in addition to microbial interaction with plant roots being important, the microbial community present in the fungal mantle plays a vital role in the fruiting process. For cultivated mushrooms, interactions with other microorganisms also occur, where bacteria present in the casing layer stimulate fruiting. Several species may play this role. In particular, studies have demonstrated the participation of Pseudomonas pituita. However, subsequent studies have shown that a mixture of Pseudomonas species or other bacterial groups is actually more effective than a single species in stimulating fruiting. According to these studies, these bacteria act in the degradation of volatile compounds that inhibit primordial formation. Dear students, let's now talk about the functions of the casing layer. The casing layer plays an ecological role simulating the conditions under which mushrooms develop in nature. However, there are several aspects of the casing layer that should be considered when establishing an appropriate environment for mushroom fruiting. Thus, the casing layer fulfills several functions. It serves as physical support for the mushrooms, keeps the environment moist, prevents desiccation of the compost, promotes the growth of beneficial bacteria, complements the water supply for the mushrooms, allows the exchange of gases between the substrate and the environment, and provides a low salinity environment. Given the many functional aspects, it is clear that there is a complex interaction network among physical, chemical, and biological variables, making it difficult to define the ideal requirements for a casing layer. Nevertheless, some conditions necessary for an adequate casing layer can be described. The casing layer must be strong and deep enough to support the growth of the mushrooms, in addition to presenting a high water absorption capacity. Additionally, the casing layer must be able to withstand frequent irrigation without losing its structure and be able to maintain good permeability to water and gases, especially oxygen and carbon dioxide. As already mentioned, the casing layer also needs to have a low salt concentration, low nutritional value, and neutral or slightly alkaline pH. Thus, the casing layer must contain calcium carbonate and have a high cation exchange capacity. Some authors have cited magnesium as a negative factor in the casing layer. However, the relevance of magnesium is still a controversial topic. Finally, the casing layer must be free of parasites and competitors. Accordingly, pasteurization or chemical treatment of the material before use is recommended. Nevertheless, after years of studies, it was concluded that good quality peat does not need any treatment. In this specific case, pasteurized peat carries fewer benefits than unpasteurized peat because of its bacterial population, which plays an important role in stimulating fruiting. However, it is important to note that for other materials used as a casing layer, pasteurization or chemical treatment are very important practices to avoid losses in production due to pests and diseases that may be present in the material. Dear students, let's now discuss about the materials used as casing layers. There is a great diversity of materials of mineral, plant and even industrial or synthetic origin that can be used as a casing layer. Among these materials, peat moss stands out as the most widely used material worldwide for the cultivation of Agaricus bisporus. It is generally accepted that peat moss is one of the best casing materials available today, and as a result, it is used as a reference in studies evaluating other materials. In addition to its water retention capacity, peat moss contains sufficient air space to allow the diffusion of oxygen, carbon dioxide and other volatile gases. Dear students, let's now talk about the mineral soil and peat-based materials. Until the middle of the last century, sterile soil or subsoil was the standard material used as a casing layer. Since then, peat has occupied this space not only because of its ideal characteristics, but also because of the ease of acquisition of this material in countries with a temperate climate. However, there is a growing concern regarding the environmental impact of the commercial exploitation of peatlands, especially in temperate and boreal regions. Given this concern, the possibility of using alternatives to peat has been explored. Naturally, the first option would be to revert to the use of soil or subsoil. In European countries such as Spain, soil was used as the base material of the casing until a few years ago, while peat was used as a structural corrective and for its water retention capacity. There is a great diversity of soils, but not all of them are viable alternatives as casing layers. Soils with a higher water retention capacity also have lower porosity and vice versa. Therefore, different types of materials have been studied as corrective materials for soil structure amendment, such as peat, sand, coconut fiber and vermiculite. Other studies, however, have addressed the use of a peat-based casing layer, which can be supplemented with alternative materials. Some of these studies will be discussed as follows, considering the two most relevant Agaricus species, which is the Agaricus bisporus and Agaricus subrufescens. Dear students, let's now talk about the casing, in which we will discuss about the casing mixture, treatment and its application. The casing is a 3 to 4 centimeter thick layer of soil applied on top of the span run compost and is a prerequisite for fructification in Agaricus bisporus. Dear students, let's now talk about the casing mixture. Covering the top of the mushroom beds after completion of the span run with a layer of pasteurized mixture is known as casing. The significance of the casing mixture is to maintain the moisture content and exchange of gases within the surface of the compost, which helps in the proper growth of the mycelium. The pH of the casing mixture should be around 7.5 to 7.8 and must be free from any infection or disease. Earlier, subsoil material or organic matter-rich soils were used as casing in button mushroom cultivation. Presently, peat is the most desirable casing material used worldwide with excellent mushroom yields and superior fruit body quality. However, peat is not available in India. In our country, casing mixture is prepared from the following ingredients: well decomposed farmyard manure, preferably 2 years old; well decomposed spent mushroom compost, 2-year-old anaerobically decomposed; composted coir pith, coir industry waste, well decomposed and water leached in the ratio of 1:1, 2:1 and 1:2 respectively, of well decomposed farmyard manure and spent mushroom compost in the ratio of 1:1, of decomposed farmyard manure or spent mushroom compost with composted coir pith, decomposed powdered bark of some forest trees, paper industry waste. Burnt rice husk is also in use along with decomposed farmyard manure in the ratio of 2:1 in the seasonal cultivation of button mushrooms in Haryana and Punjab with reasonable success. Dear students, let's now talk about the formulae for preparation of casing soil mixture. On one side, we will talk about the ingredients used, and on the other side, we will discuss about the ratio used. First, we use the 2 years old farmyard manure plus garden soil in the ratio of 3:1. Second, we can use 2-year-old farmyard manure plus garden soil in the ratio of 2:1. Third, we can use 2-year-old farmyard manure plus spent compost in the ratio of 2:1. Fourth, we can use loam soil plus sand in the ratio of 4:1. Fifth, we can use 2-year-old farmyard manure plus loam soil in the ratio of 1:1. Sixth, we can use 2-year-old spent compost plus sand plus lime in the ratio of 4:1:1. Sixth, we can use 2-year-old spent compost plus loam soil plus farmyard manure in the ratio of 2:1:1. Seventh, we can use 3 years old spent compost plus farmyard manure in the ratio of 1:1. These were the formula for using the ingredients as well as the ratio of casing mixtures. Dear students, let's now talk about the casing of mushroom beds or spawn run compost. Casing of mushroom beds or spawn run compost is necessary because casing soil is a nutrient deficient medium which helps in converting the vegetative phase into fruiting. Fruit bodies are formed in abundance and thus production is economical. It helps in conserving the environment in mushroom beds. The casing medium harbors some beneficial bacteria and activated charcoal-like material which helps in the initiation of fruiting bodies on the casing surface. The casing mixture also helps in conserving moisture in the beds and gives support to the fruiting bodies. Dear students, let's now talk about what should be the characteristics of a good casing material. First, it should have a good water holding capacity and more pore space percentage. Second, it should be capable of releasing harmful gases during cropping. Third, it should be free from harmful microorganisms. Fourth, its pH should be slightly alkaline. Fifth, it should be properly decomposed. Sixth, it should be free from heavy metals and ions. Dear students, let's now talk about the treatment of casing mixture. The casing mixture is treated either with chemicals or pasteurized with steam to kill various pests and disease propagules present in the mixture. Dear students, let's now talk about the chemical treatment of casing mixture. The casing can be disinfected with formaldehyde treatment. The formaldehyde solution is prepared by mixing 2 liters of formalin 40% in 40 liters of water to obtain 2% solution. The casing mixture made up into a rectangular pile is drenched thoroughly with this solution and then covered with a polythene sheet. The treatment should be given at least 2 weeks before casing is to be done. In other words, casing should be prepared and treated immediately after compost has been spawned. In another method, the casing soil is piled on the cemented floor and is treated with 4% formalin solution, followed by thorough turning of the soil, and it is covered with a polythene sheet for the next 3 to 4 days. It should be ensured that the casing mixture does not have traces of formalin when applied on the beds. Dear students, let's now talk about the pasteurization of casing mixture. In farms where facilities for pasteurization of compost with steam are available, casing can also be pasteurized. For the pasteurization of the casing mixture, casing soil is filled in trays, and trays in turn are stacked in the pasteurization room. Steam is introduced to bring the temperature of the casing mixture to 65 to 70 degrees Celsius, and which is maintained for 6 to 8 hours. All the harmful microorganisms, including mushroom nematodes, are killed at this temperature. Useful bacteria like Pseudomonas, which play a positive role in the introduction of fruit bodies, are not killed and survive at this temperature for 7 to 8 hours. Casing soil pasteurized in this manner gives the best result. Dear students, let's now talk about the application of casing. When the spawn run is completed, the casing is done over spawn run compost after removing newspaper sheets from the trays on racks or after opening the mouth of the polybags. Spawn run compost is slightly pressed and covered on the surface with a 4 to 5 cm thick layer of casing soil. After casing, the temperature of the mushroom house is maintained at 24 to 25 degrees Celsius for another 8 to 10 days, and water is sprayed over the casing soil. Within 8 to 10 days, white mycelium spreads in the casing soil. Thereafter, temperature of the mushroom houses is lowered to 18 degrees Celsius and maintained between 14 to 18 degrees Celsius during the rest of the fruiting period. Whenever required, watering is done with the help of a sprayer, and relative humidity is maintained at 80 to 85% throughout the cropping period. Dear students, let's now talk about the crop management. As soon as the white cottony growth of the mycelium appears on the casing surface, fresh air should be introduced inside the cropping room and bed temperature lowered to 16 to 18 degrees Celsius, which is to be maintained throughout the cropping period. The carbon dioxide level is also lowered to below 1000 ppm. Under such conditions, the initiation of fruiting bodies, that is spinning, takes place within 6 to 7 days of aeration, which reaches the harvesting stage within the next 4 to 5 days. The individual fruit bodies are harvested carefully without disturbing the adjoining pinnings and before the cap opens. The cropping period lasts for 40 to 60 days. Mushrooms appear in flushes provided optimum conditions like bed temperature 16 to 18 degrees Celsius, relative humidity around 80 to 90% by spraying water with a misty nozzle, and about 4 to 5 air changes every hour, resulting in carbon dioxide levels less than 1000 ppm in the cropping room with no light at all are maintained. Environmental factors like temperature, relative humidity, light, airflow in the cropping room, etc. all play vital roles which together determine the nature of further mushroom development. The mushroom crop grows in a cycle called flushes or breaks. Depending on the species being grown, day intervals with each successive flush bearing fewer mushrooms. These flushes normally appear in 7 to 10 days. Dear students, let's now have concluding remarks based on today's discussion. Casing and its materials play an indispensable role in the successful cultivation of many mushroom species. By providing an optimal microenvironment for the development of fruiting bodies, casing ensures consistent moisture retention, supports beneficial microbial activity, and initiates the crucial fruiting process. Selecting appropriate casing materials such as peat moss, coco coir, and vermiculite and preparing them with proper sterilization and pH adjustment significantly enhances mushroom yield and quality. Understanding the specific needs of different mushroom species and adapting the casing techniques accordingly can maximize production efficiency. As mushroom cultivation continues to evolve, exploring innovative and sustainable casing materials will be vital in meeting the growing demand for high-quality mushrooms. In essence, mastery of casing techniques is a cornerstone of successful mushroom cultivation, bridging the gap between substrate colonization and plentiful harvest. Dear students, this concludes our today's topic. Hope you have learned and enjoyed it well. Thank you.